Good day, class! I am Jomar M. Urbano, your virtual teacher in our subject, Genetics or Segs 14, and welcome to Coed TV. I hope you are all doing good and safe at home. Before we proceed with our lesson, let me remind you of the things you must have while watching this video. Number 1, notebook and pen where you can take down notes. Number 2, printed module. Make sure that you have your printed modules with you in support of our discussions and activities. And number 3, for me to monitor your responses and feedback, you must have your smartphone or mobile phone with load. You need this to send your answers via SMS to the questions that will be flashed on this video. Always be alert with this signal icon. Every time you see this, you have to respond by answering the question and sending it to my cell phone numbers flashed on your screen. And you must follow this format every time you send messages. You have to write your name, section, question number, then your answer. For example, question number 1, what is peptidoglycan? After reading the question, you can type and send your answer right away. Also, be reminded to watch the video and send your answers within the scheduled time and day for our subject. I, as your teacher, will guide you to what, when, where, and how we can tackle a specific topic. If all is set, let us now have a recap of our lesson last time. So what was our topic last time? Exactly, we talked about DNA replication. And DNA stands for... And we also talk about RNA. And RNA stands for... And for the third question, why DNA needs to replicate? I will give you 1 minute to answer these 3 questions, and your time starts now. By this time, I should receive all of your answers to questions number 1 to 3. To begin with our today's lesson, let's read this trivia. Maybe a statement you hear again and again is, over 50% of our bodies are made up of water, which is true. But did you know that protein makes up 17% of our body? All of the protein within our body has a function, and none of it gets stored. One of the best examples of protein is the antibody that fights bacteria and viruses like SARS-CoV-2, which is the virus that causes COVID-19 disease. But have you ever wondered how proteins in our body are made? To answer that question, we will now discuss the topic entitled Protein Synthesis. And here are our goals for today. Discuss the types of RNA and its roles to protein synthesis. Explain how protein is made using the information from DNA, transcription, and translation. And apply the concept of protein synthesis through the amino acids coded for the mRNA codon using the genetic code. Let's talk about first the types of RNA and its roles in protein synthesis. RNA or ribonucleic acid has 3 types. These are the messenger RNA or mRNA, ribosomal RNA or rRNA, and the transfer RNA or the tRNA. The messenger RNA serves as the intermediary between DNA and the synthesis of protein products during translation, meaning mRNA is a template that contains the message from the DNA that needs to be decoded by the ribosomes. While transfer RNA is a small type of stable RNA that carries an amino acid to the corresponding site of protein synthesis in the ribosome. Remember that amino acid is the building blocks of protein. And ribosomal RNA is a type of stable RNA that is a major constituent of ribosomes. It ensures the proper alignment of the mRNA and the ribosomes during protein synthesis and catalyzes the formation of the peptide bonds between two aligned amino acids. Remember, there are three types of RNA. Messenger RNA carries DNA message to ribosomes. Transfer RNA transfers amino acids to ribosomes. Ribosomal RNA makes up ribosome protein factories of the cell. Let's watch this video to check and strengthen your understanding about the three types of RNA and its roles to protein synthesis. Then answer these guide questions. Question number 4. What part of the cell does mRNA come from? Question number 5. What type of RNA is being decoded by the ribosome? And question number 6. What do you call the building blocks of protein carried by tRNA? Now, let's watch this video.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
Okay, this time I will give you 1 minute to send your answers. Let's now proceed to genetic coding. And these are the things you have to remember. Combination of three nitrogenous bases will produce a codon, and a codon designates an amino acid. An amino acid may have more than one codon. There are 20 amino acids but 64 possible codons. Some codons tell the ribosome to stop translating. To easily understand this, let's use this amino acid identifier chart. This can also be found on your module Figure 1.6, page 6. Use the code by reading from the center to the outside. Example: the codon AUG codes for methionine. Another one: CUU, CUC, CUA, and CUG will give you an amino acid called leucine. As you observe, the amino acid leucine may have more than one codon, and it also happens to other amino acids. Now, it is your turn. For question number 7 to 10, what amino acid can be decoded to codon? UCA, CAU, GCA, and AAA. I will give you 2 minutes for this. Your time starts now. Time's up! I should receive all of your answers to questions number 7 to 10. Let's now talk about the two major processes involved in protein synthesis. The first one is transcription. It is the process of copying the sequence of one strand of DNA producing mRNA. And the other one is translation. In this process, the mRNA is being decoded into a polypeptide chain to become a specific protein. So what happens during transcription? First, RNA polymerase binds to DNA and separates the DNA strands. Second, RNA polymerase then uses one strand of DNA as a template to assemble nucleotides into RNA. Third, specific base sequences act as signals to stop. And lastly, mRNA leaves the nucleus through its pores and goes to the ribosomes in the cytoplasm of the cell. How about during translation? Ribosomes read mRNA 3 bases or one codon at a time and construct the proteins. And then, a sequence of amino acid bonded together by peptide bonds. After transcription and translation, there are 3 other steps to complete protein synthesis. Step 1. Initiation. In the cytoplasm, protein synthesis is actually initiated by the AUG codon or the amino acid methionine on mRNA. The AUG codon signals both the interaction of the ribosome with mRNA and also the tRNA with the anticodons UAC. Step 2. Elongation. During the elongation step, the polypeptide chain adds amino acids to the chain protein grows as the ribosome reads the mRNA. And the last step, step 3, termination. Termination occurs when a nonsense codon, UAA, UAG, or UGA is encountered. These codons are also called stop codons. And these are all the processes involved in protein synthesis. So for question number 11 and 12, question 11, if a person lacks amino acid methionine, what do you think will happen? And question number 12, what are the 3 stop codons? I will give you 3 minutes to answer this. Your time starts now. By this time, I hope you've already sent your answers. For your extension activity, go to pages 11 to 13 of your module. And let me read the direction. Apply the concept of protein synthesis using the genetic code chart. Study the given scenario and analyze the profile of the suspects. Then answer the following questions. So here's the scenario. An intruder raised the biology laboratory of the College of Education. The college is in chaos. The university administrators and faculty demanded to know the culprit. They have to identify the culprit and do it fast. They had 3 suspects, namely Rudy T, Von Go, and Rambo Tan. As a forensic expert, your mission is to identify the intruder using the profile of the suspects and other information taken from the laboratory. You have to submit your answer before our next meeting. And to end our lesson, let us read this quotation from Robin Sharma. You know you're winning when you see you're being copied. That's all for today. See you again for our next Coed TV. Keep safe and God bless us all.